Hey guys, I wanted to go over this Plasmid question that I posted. It's from an old exam. You can see 2007. So you don't need to worry about actually completing this question. I just want you to follow my logic through this video. The reason why I'm doing this is because this really forces you to think through gel electrophoresis and plasmids, these new topics I'm bringing up today, in an important way. So this is not about you being able to actually solve this puzzle in this question yourself. Just follow this through and hopefully you learn about plasmids and gel electrophoresis techniques from it. So the student is told they're given this plasmid diagram down here. So I'll draw that here on my whiteboard. So here's my plasmid, a circular loop of DNA. And then I just wanted to say a couple things before I continue. One, I just realized in my last question that my last video, the origin of replication, an ORI region on a plasmid, I should have said more clearly, that would be where DNA polymerase would attach to replicate the plasmid, because bacteria do replicate their own plasmids when they divide. And that is important. So that would be the origin of replication and refers to DNA replication. That's just sort of an arbitrary beginning and end point to the plasmid. We don't need to worry about an ORI region in this problem. We're told that, the other thing that's sort of a background side note is if you have a closed loop of DNA, how many pieces do you get if you cut it once? You get one piece. Imagine this loop opening up into a linear piece. If you cut it twice, you get two pieces, right? That's different with linear DNA. If you have linear DNA, and you, like eukaryotic DNA would be, and you cut it once, that'll produce two pieces. If you cut it twice, that'll produce 1, 2, 3 pieces, and so on, right? So that's just an important difference between plasmid and eukaryotic DNA. So the student is told here that the plasmid is 100 kb. That means 100 kilobases or 1,000 bases. When it is cut with the enzyme EcoR1, it produces two pieces. So that must mean that there's two restriction sites for EcoR1, and we have a marker lane here. They call it the molecular weight standard. It's the marker lane, right? And they're fragments of known sizes. So when we digest this plasmid with EcoR1, we get one piece that's 70,000 bases long, that's 70 kb, and we get a second piece that's 30 kb. The digest with Hay3, this is a separate digestion reaction that produces also two pieces, so there's got to be two Hay3 sites on this plasmid. And again, the site means where the enzyme cuts, and it'll produce a 60 and a 40 kilobase pieces. And then they do something interesting here. They do a double digest. They put EcoR1 and Hay3 in that tube with the DNA together, and it produces this banding pattern here. So you've got to produce a piece that's 40, 30, 20, and 10 kilobases. So how do you do this? Well, again, don't worry about actually doing it. Just follow my logic through. So I know there's two EcoR1 restriction sites. So let's make one there. Doesn't really matter where you start. And let's make one here because I know that I've got to produce a 70 kilobase fragment and a 30 kilobase fragment. So there's my solution for EcoR1. If I cut with EcoR1, slice, it produces a 30 piece and a 70 piece, and that produces those bands when it's run on the gel. If I cut with Hay3, it produces 60 and 40. So I could do a similar, I could have my Hay3 cut be basically the same, right? And produce 40 and 60. But this gets tricky because when I double digest them, I have to produce 40, 30, 20, and 10. So the answer to this, I had to play around with this a bunch, is one Hay3 site is going to be here. So here's Hay3. So that's cutting it once. And then I'm going to put another Hay3 site around here because the first thing I have to make sure of is that I'm cutting it 40 and 60, right? So this piece right here between this and this is 10. This whole piece is 30, but this little piece now is from here to here is 20. From here to here is, I think 40. I'm gonna have to double check this. And I think this is then gonna be 30. So do I have my Eco R1 set? 30, 70. Do I have my Hay 3 set? 20 plus 40, 60 and 40. And do I have the double digest set? It's going to produce a 10 piece, a 20 piece, a 30 piece, and a 40 piece. Boom. This is the answer to that question, right? But again, I don't care about you getting that right yourself because it's from an outdated exam. It's just more that you're following me through that I can check my answer against this and then it makes sense. This was a really hard question. It says here in B, describe how recombinant DNA technology could be used to insert a gene of interest into a bacterium. Okay, well, recombinant DNA is when DNA from two species are mixed together. So what I would need to do, let's say we're taking DNA from a eukaryote, for example. Let's say I had some chimpanzee DNA or something. I would need to cut out a chimp gene using the same restriction enzyme that I cut this plasmid with. So let's say that enzyme is Hay3. If I cut my chimp DNA with Hay3 and I cut this with Hay3, the plasmid would open up at that point, and then there would be sticky ends that I could use to anneal, or I could say ligate, those fragments together. So here's my chimp. Oh, wow, that marker is no good. Here's my chimp DNA coming in there, right? So what I did was I cut the chimp DNA with the same restriction enzyme that I cut the plasmid with. And that should be sufficient for your answer. Now, keep in mind here that this plasmid would now no longer be 100 kilobases because I inserted some extra DNA into it. Let's say this was 20,000 base pairs long. So now I have a plasmid that's 120 kilobases. So if I get a recombinant plasmid here in my experiment, it's going to be a bigger plasmid than if I did not get the gene inserted and did not make the recombinant. So this question here about how could the recombinant bacteria be identified? Well, there's several ways to do this, and I'm going to make some separate videos going over some other ways. But one way we could say to get this credit here is that we could run gel electrophoresis on this DNA. And if we got a band from cutting this once, if we got a band that was 100 kilobases, we would know the plasmid didn't take up the new gene. But if that fragment was 120 kilobases, we would know that the fragment was taken up. So that would be one way we could check to make sure that the bacteria had been transformed, right? And then how could the expression of the gene of interest be ensured? How can I make sure that the bacteria are actually producing this chimp protein? Well, there's a few things to discuss here, right? Number one, I think you only need to say one thing. Number one, and this goes for any type of transcription question. In order to get transcription to happen, you need a promoter region. That's where RNA polymerase attaches. So you would need some region here in front of the gene that would be a promoter, right? And that's just a general guide. That's an important note for all of you guys, right? This is a general note for all types of transcription questions. You need a promoter region. Otherwise, the bacteria won't even see this gene. Think of the promoter as being like a flag, being like, hey, transcription proteins, transcription factors, RNA polymerase, here, here, here. This is where we want you to be. So we do have to have a promoter region. Now, it's possible that the biologists know specific promoters and could actually synthesize DNA to have a specific sequence that they could put there. So let's say we needed the sequence to be TTACTATTT. A lot of times there's a lot of T and A's in a promoter region, like TATA, TTTA, TEA. So we could have that sequence put there. You don't have to explain how to do it, but you should just know that like scientists can make any DNA sequence. They can just synthesize it. If you want a DNA strand that goes GTA, 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 a geneticist could produce it. So we've synthesized a promoter that goes in front of that gene. The other thing, and this is the last point I'll make in this video, I think, but kind of tricky here. But remember, eukaryotic DNA has introns and exons. Remember, introns get removed by eukaryotic cells before a translation. Exons get expressed. Bacteria don't have those. So if you just chopped the chimp gene and put it in there, the bacteria wouldn't know how to transcribe and translate that gene because of the introns. That would confuse the bacteria. So what you would need to do before inserting this chimp gene is cut out the introns. And there will be another video coming that says how that's done or a way that's done, but that's another thing we could discuss here is we would remove the introns from the eukaryotic gene so that this prokaryote could express it. The final question on this is discuss how a specific genetically modified organism might provide a benefit for humans and at the same time a threat. I think I'll discuss this. My concern about GM technology is not huge for you guys on this year's exam, but I'll have a separate discussion of that at some point. Okay, bye.